is open again. We are closed for a few days, but now we are fully open. Our current exhibit, Dinosaurs Revealed, is open through September. There are 26 animatronic dinosaurs, dozens of fossils, plus 2 full dinosaur skeletons. Advance tickets are highly recommended as we are sold out for the coming weeks. You can also catch a show at the planetarium, the Empirical Theater, or tour the submarine. Check out our website for the most up-to-date information and to view our COVID safety guidelines. I hope you enjoy tonight's pre-pub trivia and the music by local Portland artist Trinetti. Putting on these live shows takes a lot of work, and we have an amazing partner that helps make that happen. So a big shout out and thank you to Celestream for providing the live streaming service for tonight's Science Club. We really appreciate their support. All right, join us for our 9th virtual OMSI After Dark Best Of. We have found so many delicious products from Oregon that we want to celebrate with the event by honoring the best beers and ciders, seltzers, and snacks from the past 7 of our virtual OMSI After Darks. Tickets include 10 items and access to the virtual event. Visit omsi.edu/bestof for all the details. We also have virtual workshops that you can join us at. You can learn to make cow's fresh milk mozzarella cheese or a personalized batch of kraut or kombucha. Visit omsi.edu/workshops for more information. We also want to help put the pub back in the Science Pub experience by ordering some delicious food and tasty beverages from one of our partners around the state. We've got plenty of places that are supporting us from Portland, Salem, Eugene, Corvallis, and Bend. All right, so for tonight's event is set up like our regular Science Pub program. For those of you who don't know what that looks like, we're going to begin with a habit-themed trivia. That is a warm-up for tonight's talk. So go ahead and grab a pen and paper so that you can participate. After that, we'll have a lecture by Russ Poldrack from Stanford University. After the lecture, we will take your questions. You can submit them at any point during the lecture via the comments in our live feed on Facebook and YouTube. We'll collect them all, and after the lecture, I will ask your questions to our speaker. If you enjoyed tonight's lecture, please consider making a donation or purchasing a Science Pub pint glass. But don't worry, there is no pressure to donate. Our mission at OMSI is to inspire curiosity by creating engaging science learning experiences for people of all ages and backgrounds. So sit back, relax, and get ready for tonight's great lecture right after our trivia. All right, so I hope that you grabbed your pen and paper. This week we have a special guest, Melody Beard. She is one of our awesome events managers, and she has come to play along live with you at home. So please give her a warm welcome.
Hello.
Welcome, Melanie. It's so great to see you. How are you?
I am good.
Are you excited to learn something about habits?
I am. I'm hoping to learn something new, either something I can learn how to make a good one, a good habit, or how to break a bad one. So I'm very excited.
Awesome. Me too. So tonight we're going to encourage you to play along at home. Maybe you're going to wager who does the dishes tonight, or who has to vacuum later. I already did the vacuuming at my house, so that's off the table here. But get ready, there'll be 10 questions. I'm going to read out a question, give you some time to guess, and then reveal the answer before moving on to the next one. Are you ready, Melanie?
Yes, I am.
Okay, let's go to our first question. All right. Why did William James travel to the Amazon in the late 1860s? Was it A, to study the habits of native Amazonian people. B, to search for cures for his ailments, for his many physical ailments. C, to bring back samples of Amazonian plants and birds. Or D, to search for psychedelic drugs.
Well, okay, so just thinking about tonight's topic, I am going to go with B, to search for cures for his ailments.
Interesting, even though they're physical ailments?
Well, that's true, but I don't think he was trying to bring back, well, I don't think he was looking for drugs. Maybe he was wanting to bring back plants and animals, but I'm going to stick with B.
We're going to stick with B. All right, let's see. The answer is B, to search for cures for his many physical ailments. You were right. Nice job. All right, let's see. What's next? Which organism has the highest brain to body size ratio? Is it A, ants, B, humans, C, lions, or D, sharks?
Well, I mean, I want to say the obvious answer. Well, not the obvious answer, because I don't know. These are very weird. Like, I do not know the body to brain size ratios of these animals, but I think I'm going to go with humans.
That'd be nice, right?
That'd be nice.
Yeah. We like to think that at least. But in fact, it is ants. 1 to 7. So those go right in order. Sharks seem to have the tiniest brain to body ratio. Ants have the biggest. Ants are quite amazing.
Ants are amazing. Wow.
Yeah. Strong and smart, maybe. All right. Number 3. How many types of dopamine receptors are there in the human body? A, 1, B, 5, C, 15, or D, 26?
These are the feel-good receptors. I
think so.
I'm gonna think so. I'm gonna think that there's probably more than one.
Okay.
But the range is kind of high. Let's go for C, 15. 15.
All right. Nice middle number there. But the real answer was B, 5. A, B, C, D, E? I don't know. We'll have to find out more. All right. Question 4. Where is Phineas Gage buried? Is it A, Boston, Massachusetts, C, Santiago, Chile, B, C, Cavendish, New Hampshire, or D, Colma, California?
Well, I really have no idea, but I am just going to go for the West Coast, because also I think our speaker is from California, so maybe this has some correlation to why he knows about this, so I'm going to say D.
Colma. I haven't been to Colma, but it does say railroad construction, so that's probably a good guess. Yes, D, Colma, California. Excellent. Looks like he only has one eye open too. What is the part of the brain where strokes have been known to cause people to forget they are a smoker? Ooh, that's convenient. A, the insula, B, the basal ganglia, C, the subthalamic nucleus, or D, the prefrontal cortex?
I thought I kind of knew parts of the brain, or I guess I only know the major ones because I don't really know any of these except for 2 of them. I'm going to guess it's not the prefrontal cortex. It's probably one I've never heard of. I'm going to go with A.
The insula? All right, let's find out. You're right. Nice job. It is the insula. We'll have to find out where that is in our brain.
Exactly. Yeah, I'm curious.
Yeah. Okay. Number 6. Which organism was essential in the discovery of optogenetics? Was it A, electric eels, B, green algae, C, poinsettias, or D, voles?
I've only recently learned about optogenetics, but I don't remember this one. I'm gonna go with voles, just because I know there's been some research done on voles, so that's my guess.
All right, voles, were you the one? No, somehow it was green algae, B. That's quite a surprise.
Maybe organism should have been the clue there, but anyways, after I think about it, that's interesting. Now I want to learn about that.
Maybe it has to do with branching patterns or something. I don't know anything about optogenetics yet, so we'll find out. All right, 7. Which of the following celebrities is known to have the habit of biting their nails? Hopefully they have some close-ups on their fingers. Is it A, Tom Cruise, B, Emma Stone, C, Jennifer Lopez, or D, Will Smith?
Oh, this is a hard one too. I have no idea. I know that Tom Cruise is kind of a hyper person, and that's the only thing I know about any really of these, so I'm going with A.
Tom Cruise? Okay. That Oprah episode really made an impression on people.
It really did.
It is Tom Cruise! You had it right. Guess they don't have close-ups on his hands. Maybe they have fake nails on him. All right. Number 8. What book originated the idea that it takes 21 days to form a new habit? I've definitely heard this one. I don't know anything about it. Is it A, Cybernetics by Maxwell Maltz, B, The Behavior of Organisms by B.F. Skinner, C, The Power of Habit by Charles Duhigg, or D, Metaphysics by Aristotle?
I have not read any of these books. So I'm just going to go with the classic answer of C.
C, yes. The SAT's favorite. No, it was A, Psycho-Cybernetics by Maxwell Maltz. I am sorry.
Yeah.
We'll be doing some reading later.
Yes.
All right, number 9. Which plant has the highest natural level of dopamine? Let's be going on some shopping lists later. Is it A, tomatoes, B, bananas, C, avocados, or D, spinach?
This is also one that I really have no idea about. But what would be the most addictive food possibly? What do you want to keep eating? I'm going to go with bananas.
All right.
They're sweet. I eat a lot of them.
Chimpanzees seem to like them.
True.
Who knows?
Hey!
B, bananas. Wow, good instinct there. Nice. All right, grab a bunch today. All right, last question. Hope you're doing well, everyone out there. What year was Chicago's song Hard Habit to Break released? Was it A, 1969, B, 1975, C, 1984, or D, 1990?
Well, I do love me some Chicago, but I don't know this song, or I don't know the name of the song.
Sure.
At least they're broken up by decade. So I'm gonna say B for 75.
1975. All right, let's find out. Oh, I'm sorry.
Oh.
It is 1984.
Well.
Good try. You did pretty good though. Nice job.
Yeah, thanks.
Yeah.
Nice. Well, I'm excited to learn more tonight, so.
Yeah, hopefully we'll get some more answers very soon. So thank you so much. And we'll see you again soon.
Bye.
Bye. All right. Excellent job, everyone. Thank you for participating in our trivia. So now I would like to introduce our speaker for the night. Dr. Russell Poldrack is the Albert Ray Lang Professor of Psychology at Stanford University. He runs the Poldrack Lab that is based in the Department of Psychology at Stanford University. His lab uses tools of cognitive neuroscience to understand how decision-making and executive control and learning and memory are implemented in the brain. He is the author most recently of Hard to Break, Why Our Brains Make Habits Stick, as well as The New Mind Readers, What Neuroimaging Can and Cannot Reveal About Our Thoughts. He lives in San Francisco and can be found on Twitter at Russ Poldrack. Please join me in welcoming Dr. Poldrack. All right, take it away, Russ.
All right, thanks very much. It's a pleasure to be here and talk about my new book, Hard to Break. Nice job on the trivia, by the way. Those were especially hard questions, I thought. So often when people think about habits, they have to jump right to either thinking about sort of good habits, like the stuff that we'd like to do, but turns out to be hard to do, or bad habits, the stuff that we feel the need to do, but often wish we didn't do. But one thing that's important to realize is that there's what you might think of as an entire zoo of habits that inhabits our lives and our behavior from day to day that sits in between those, and we'd really be lost without all of these habits. And so I want to spend a bit of time talking about first why it is that habits are so important. So first, think about your morning routine. Mine involves getting out of bed, going into the kitchen, turning on the espresso machine, wait for it to heat up, grind some beans, make my coffee, and then get some breakfast and go sit down and check my email. What's really astounding is how very rarely I actually think about any of those things, right? I don't think, oh, I need to now turn left to go from the bedroom into the kitchen, or I need to press that button to turn on the coffee machine. I just do these things because they're routine. And this is what habits do for us. They basically allow us to sort of store these kinds of routines so we don't have to think about them. Now, William James was mentioned in the trivia, and the reason that he was included there is because he's probably certainly the first kind of psychological writer about habits, and probably wrote about them in a more compelling way than almost anybody has. This is a quote from his 1890 book, The Principles of Psychology, talking about why habits are so important. He says, "The great thing then in all education is to make our nervous system our ally instead of our enemy. For this, we must make automatic and habitual, as early as possible, as many useful actions as we can. There's no more miserable human being than one in whom nothing is habitual but indecision. Full half the time of such a man goes to the deciding or regretting of matters which ought to be so ingrained to him as practically to not exist for his consciousness at all." And we've all known that person, right? Just points out how important habits are for us. Now, one way to think about why we have habits and why they actually in our brains rely on a different set of systems than other types of memory is that there's a kind of a fundamental tension between the kind of things we have to learn and remember out in the world. Certain things in the world change often. They change all the time. So think about driving to work. If you drive to work and park your car in a parking garage, unless you're lucky enough to have a dedicated spot, you have to remember every day where you parked your car. And, I used to have to do this years ago, and I would regularly end up walking up and down a couple floors looking for my car because I didn't remember where I parked it. So some things change all the time, and we just have to sort of remember those. But other aspects of the world kind of stay the same, right? The location of the pedals in our car, what those pedals do, they don't really change from day to day. And so there's no need for us to have to think about them. We should just, have our brains kind of record them. And that's exactly what the habit system in our brain does. It basically records the things in the world that don't change so that you don't have to think about them. And so that they'll be sort of turned on automatically when the right time comes. So let's talk a bit about where habits actually come from in the brain. And if you go to the internet, you might think that they come from this thing people call the lizard brain. These are actual headlines from stories on the web. How your 200 million-year-old lizard brain is holding you back. Don't listen to your lizard brain, quieting the lizard brain. So the idea is that, we have this kind of prehistoric part of our brain that we share with lizards that basically is, just built around sort of routine behavior and just acting on impulse. So the idea is, the lizard brain sees a bug, wants a bug, eats a bug. And without really thinking about it, they just are sort of creatures of habit. These ideas were first written about in the 1970s by a neuroscientist named Paul Mclean, who actually had a whole laboratory where he studied lizards. Now, interestingly, the parts of the brain that people are talking about here largely focus on a set of brain regions that you heard about on the trivia called the basal ganglia. And I'm highlighting a few of them in the middle here. This is actually my brain. That's a slice through my head. And this shows like deep in the middle of the brain, these different brain regions. I'm not going to make you remember the names of, but these are all part of that dreaded lizard brain. Now, it turns out that the whole idea that like the lizard brain is sort of fundamentally different from the brains of mammals or humans has sort of been debunked by neuroscience. It turns out that pretty much all vertebrates have roughly the same plan for how their brain is organized. We all have a cerebral cortex. We all have these basal ganglia. Obviously, we have a lot more cerebral cortex than a lizard does, but our brains are not really qualitatively different from a lizard brain. We just have more of certain things. So in order to understand like why the basal ganglia are important for habits, we have to step back a bit and understand their role in sort of helping us choose what to do in general. So I want to walk you through, what we know about the neuroscience of how we choose the things we do in the world. So let's say you're driving down the road, and in this case, you're in England, so you're driving on the left side of the road, and you come to a fork in the road. And you have to decide, do I go left or right? And there's a couple of major players in this decision. One is the cerebral cortex. This is the kind of, crunchy candy shell on the outside of our brain. And then we have the basal ganglia kind of down here in the middle. And when you go to make an action, the way that we think this works is you have different sets of neurons out in the cerebral cortex that basically are kind of voting for the different actions you might take. You might have one set of neurons saying go left, you might have another set of neurons saying go right. And those neurons both basically send their inputs down into the basal ganglia and sort of fight it out. They basically try to figure out, which one of them has a stronger effect on the neurons in the basal ganglia and ultimately wins. And then what we know is that the same neurons that go in basically get connections that come back. We refer to these loops in the brain that go from the cortex down to the basal ganglia through some other places and then back to the cerebral cortex. So the idea is that there's this competition between, whenever we go to decide what to do, there's this competition, and the basal ganglia plays a critical role in kind of, figuring out who wins that competition. Now, what determines who wins? Well, one thing is, do we know one of those actions to be good or not? And psychology doesn't have a lot of laws like physics does, but one of our laws is this thing called the law of effect, which was coined by a psychologist, Edward Thorndike, more than 100 years ago. And the law of effect is something that is obvious to everybody who's listening, which is basically if you do something and it leads to a good outcome, you're going to be more likely to do that thing. If you do something and it leads to a bad outcome, you're going to be less likely to do that thing. So if you go left and you get the thing you wanted, the rainbow at the end of the road, you're more likely to go left next time. If instead you hit a roadblock, you're less likely to do that the next time. So how does that work in the brain? Why is it that certain things happen more often than others? This is where our friend dopamine comes in. Von Bell is a writer about neuroscience who said that dopamine is the Kim Kardashian of molecules. Everybody has heard of dopamine. Everybody thinks they know what dopamine does. And it turns out that dopamine is incredibly complex, and both in our brain and the rest of our body. It turns out that dopamine isn't primarily about pleasure. It's really about wanting. Neuroscientists distinguish between wanting and liking, and dopamine is more about wanting than liking, and other neurochemical systems are actually more about liking than wanting. I talk about that some in the book. So let's look at what happens to dopamine when we go through the world and engages in these actions, to try to understand why it turns out to be so important for habits. Now, this is just a kind of a picture of, this is a slice down the middle of the brain. This is just showing like down in the middle of the brain, really deep, basically down in the, almost in your medulla oblongata, is where the neurons live that kind of send out dopamine to the rest of the brain. They send it all over the place. This is just kind of showing where some of those places are, but the basal ganglia over here in the middle receive, by far the strongest connections from the dopamine system. They're getting a ton of dopamine compared to other parts of the brain. So let's see what happens. Let's see how dopamine responds as you go through the world, making your decision at that fork in the road. So you come to the fork in the road for the first time. And this is, I'm just showing you a few dopamine molecules here to highlight the fact that, the dopamine neurons in our brain are always firing. They fire maybe 5-10 times a second, even when you're kind of, doing nothing. If they didn't fire, you'd be in trouble, because you need some dopamine around all the time, or you would become catatonic. There was this idea in Silicon Valley last year about the dopamine fast, which, really didn't make very much sense biologically. So that, there's a little bit of dopamine that's always sort of hanging around. And so let's say you decide to go left, and you get that rainbow at the end of the road. Now what happens is dopamine neurons fire a lot. They send out a ton of dopamine into the basal ganglia. That's what I'm showing here with a bunch of dopamine molecules. Okay, so that's what happens the first time. Now, you might think that means, oh, hey, that means dopamine really likes rewards. It turns out to be a little more complicated. Let's say you've done this 100 times. You've come to that intersection 100 times. You've always gone left. You've always seen the rainbow. What's going to happen now is the dopamine neurons are actually going to fire strongly when you get to that fork in the road, because that's the thing that now tells your brain, oh, hey, you're about to get a reward. So it's not so much firing when you get the reward, but firing to anything in the world that kind of predicts. One way to think about it is that predicts the world is better than you thought it might be. Then when you actually get to the rainbow, the dopamine neurons just kind of do their regular thing. They don't really get excited anymore, because that was predicted. So what they really do is they fire when you have an unexpected signal that something good is happening or is going to happen. Conversely, if you have, if the 100th time you suddenly have a roadblock, now they're going to get really quiet. They're going to hardly fire at all. So the idea here is that dopamine neurons don't necessarily respond to reward. They respond to what we call reward prediction error. That is, when is the world better than you thought it would be, or worse than you thought it would be? And this is a really interesting idea, and I go into this more in the book, because it relates to a set of ideas that have been developed in computer science and artificial intelligence from researchers who wanted to understand, like, how should any system that wants to go learn by trial and error out in the world, how should it go about learning? And it turns out that any such system has to basically compute this thing that we call a reward prediction error. So it provides us with a really strong theory for trying to understand, what it is that dopamine does when we learn. So now, how does this actually, ingrain habits? Well, it turns out that dopamine plays a very specific role in the basal ganglia, which is to help basically cement the relationships between cells that are firing together. So there's an idea, people often say that cells that fire together wire together. The idea being that when one neuron causes another neuron to fire, in general, that's going to cause the connection between them to get stronger. In the basal ganglia, this is true, but it also requires dopamine to be present. So the idea is that, let's say this is a neuron coming into the coming into the basal ganglia from the left, and this is one of these go left neurons that want you to go to the left. So that neuron fires, it sends some neurotransmitters over, they hit this other neuron, this neuron fires and causes you to go left. And let's say this is the first time and you get that unexpected reward here, the rainbow. That's going to cause some dopamine to get output into your basal ganglia, some extra dopamine there. And what that's going to do is cause a change such that next time, the connection between these two neurons gets stronger. So it's easier for this neuron to make this neuron fire, and ultimately easier for it to make you take the left turn. So why are some things so likely to cause habits? Well, one thing to keep in mind, right, is that our brains evolved to respond to natural rewards, the kind of things we find out in the world, like, fruits that grow on trees. And, in evolutionarily, we might have gone and picked some oysters out of the ocean, or gone and hunted down an animal and eaten its bone marrow. Those are things that can be tasty, right? But compared to the stimuli of the modern world, our brains are basically just sort of ill prepared, right? And particularly when it comes to drugs, particularly like methamphetamine or cocaine, that basically directly turn on our dopamine system. Pretty much every drug that people get addicted to has either a direct or some kind of indirect effect on the dopamine system. We know that, the kind of ultra processed, highly palatable food that humans eat increasingly also has strong effects on the dopamine system. And it's still up in the air in terms of whether, social media and digital devices really drive differences in dopamine response, but there's reason to think that happens as well. So that's part of the reason why, we're increasingly, sensitive to these types of stimuli, which are really good at generating habits. So now I want to turn to asking, why are habits so sticky? We all know, anybody who's tried to ever change their behavior knows that habits can be incredibly sticky. And I want to talk about a way of thinking about habits that kind of helps understand this, that has largely come from studying rats, basically doing things in cages and getting food for doing it. So let's take a rat who, is hungry. And this rat has learned that, if it goes and puts its nose in this little particular spot in its box, then some food's going to come out in the hole on the left. And here, the rat's hungry, it goes and gets his food and then eats it. Now, you might think, why would you do something if you weren't hungry? Well, it turns out that when we form habits, those habits ultimately become sort of unmoored from the goals that originally drove us to do the behavior. And one way we can see this is by basically devaluing the goal, making you not want it anymore. So let's say you're a rat and I give you so much cheese that why would you want to go eat some rat chow now? You're so full of cheese. And so now what we can do is put you back in your box and see, do you go stick your nose in the hole? If your behavior was, in service of getting the rat chow, then you're just going to chill. You're not going to go stick your nose in the hole to try to get rat chow. But if your behavior is a habit, by which we mean it automatically occurs whenever you end up in that situation, then you're going to go do the thing even if you don't want the food. And this is exactly what happens when rats are exposed to these kinds of situations. It's harder to test these sort of things in humans. And that's, I can't bring somebody into my laboratory and develop the kind of habits that people can do when they study rats. And so that's why a lot of what we know actually comes from the study of animals like rats. But nonetheless, we think that the similar types of things happen in humans as well. Another reason that that habits are so sticky is because they basically become mindless. They become so automatic that we don't think about them anymore. This is a road through the South Island in New Zealand. Incredibly beautiful, incredibly remote. And back about a decade ago, my wife and I were on vacation. We were driving from Christchurch up to Marlborough, up in the top of the South Island. And I had never really driven on the left side of the road before, but I was doing pretty well, I thought. And so we're driving along one of these roads and we come up to some construction, and they basically made us get on the right side of the road. And it was one of these things where there's a traffic light that basically says wait and lets all the cars from the other direction come, and then lets you go. There's very few cars out here though. It's really in the middle of nowhere. So we wait for our time, and then we get on the, we get in the construction zone, and we're driving in the right lane, in our right lane, right? And we go like that for a couple of miles. The construction ends, but I'm so in the zone that I don't even realize that it's ended and there's no other cars around. I keep driving in the right lane for at least a couple of miles before we basically almost have a head-on collision coming around one of these corners. Fortunately, everybody was going slow. But it just highlights the fact that, that once that our habits become really sticky because we just don't think about them. They just sort of happen. Another thing that we know about habits that has also been learned mostly from studying rats, but then, as I'll show you, confirmed in humans, is that old habits, even when we think we've gotten rid of them, they often come back with a vengeance. And, this is certainly seen in, people with drug addictions who, relapse many, many years later. What we've learned from this research is that basically, when you first learn a behavior in a particular context, that behavior is always going to be the strongest behavior in that context. So in some ways, it becomes sort of the default. It's like, all else being equal, that's the thing you're going to do in that situation. And so anything that you learn later, including like, turning off the habit and doing something else, becomes learned in a much more kind of context specific way. You learn it as like a specific reaction to a particular situation rather than kind of your new normal. Here's an example. Hopefully no spider phobics in the audience. Sorry to freak you out. This is a tarantula, obviously, and Michelle Krask, who's a psychologist at UCLA, did a study a few years ago. She's interested in anxiety disorders and wanted to look at the treatment of spider phobia. So she brought in people who claimed to be afraid of spiders and put them through a whole procedure to try to basically get rid of their spider phobia. And it started with like just seeing the thing across the room, and then slowly getting closer and closer. And by the end of the training, they basically would let the tarantula walk on their hand. So then she wanted to see kind of what happens when she brings people back if she either put them in exactly the same context, like the same person, the same room, or if she put them in a different context, different room, different person they're interacting with. And it turned out that the people who were put back into the same room were less afraid of the spider than the people who were brought back and put in a different room. And the idea is that, their ability to kind of, override that fear of the spider became really closely tied to like the specific room and the specific building, the specific person they were interacting with. And this is why we think that, treatment for these kind of phobias is so difficult. They often come back because those fears become kind of the default in any particular situation. Now, we know that overriding or changing our behavior requires us to exert control. And so now I want to turn to talking about the part of the brain that we know to be most responsible for our ability to exert control. And that's the prefrontal cortex. You could think about it as the brain's C-suite, where the executives sit. You heard about Phineas Gage earlier. This is Phineas Gage. He's probably the best known person to have had damage to his prefrontal cortex. He was working on the railroad in Cavendish, New Hampshire in the 1860s, doing what they call tamkin, which is basically taking this big metal rod and tamping down explosives in a hole. Unfortunately, the explosives went off and basically propelled the rod through his head. The reason that his eye is closed is because his eye is gone. So basically the rod went up through his head, out his frontal lobe here, Amazingly, he lived. He had terrible infections, but ultimately was able to survive this horrendous injury, and ended up going back to work and being able to at least function somewhat reasonably as a human. But something was very different about him after his injury, and this is from a report by his physician titled "Recovery from the Passage of an Iron Bar Through the Head." This is the quote: "The equilibrium or balance between his intellectual faculties and his animal propensities seems to have been destroyed. He is fitful, irreverent, indulging at times in the grossest profanity, which was not previously his custom, manifesting but little deference for his fellows, impatient or of restraint or advice when it conflicts with his desires, at times pertinaciously obstinate, yet capricious and vacillating, devising many plans of future operation which are no sooner arranged than they're abandoned in turn for others more appealing. In this regard, his mind was radically changed, so decidedly that his friends and acquaintances said he was no longer Gage." Now, another way that we can try to understand the role of a particular brain system is to look at differences in species, across species, in terms of how much of that brain structure they have. This is my cat, Coco. And Coco is a wonderful cat, but no one would ever accuse her of having any degree of self-control. She really likes to chew on electrical cables. The thinner, the better. I can't tell you how many charging cables for laptops she's ruined in the last, however old, 12, 13 years. But this is the part of the reason why Coco has so little self-control is because she has so little prefrontal cortex. This shows the relative amount of prefrontal cortex across a number of different species, from humans down to cats. And what you can see is that humans have a really substantial amount of prefrontal cortex, actually not clearly that much more than other apes, though ours is clearly organized differently. And we think that it's this substantial amount of prefrontal cortex that is what allows us to engage in the kind of self-control that we think is important for stopping ourselves from doing things. Now, that's a particular function that my lab's done a lot of work on trying to understand. And what we do is we bring people into the laboratory, we put them in MRI scanners and look at their brains while they do things. And one of the kind of things we do is sort of a laboratory analog to the following, which is, you're standing at a crosswalk, the light turns green, you start to walk, and then suddenly you realize there's a car coming. You have to very quickly stop and get out of the way. We call this response inhibition, the ability to stop oneself. And we and others have worked out, what the circuitry is in the brain that involves both the prefrontal cortex and the basal ganglia. In some sense, there's sort of a short circuit mechanism within the basal ganglia that lets you very quickly stop what you were going to do and potentially do something different. Now, you might think that it's this type of sort of ability to suppress a behavior or stop ourselves that's really important for ultimately being able to kind of, get rid of bad habits and change our behavior. And people often think about this in the context of willpower, right? And even if you ask healthcare experts why some of their patients have trouble, losing weight or other, addressing other types of health problems, they'll often say, well, I don't think they have very good willpower. It turns out that willpower probably doesn't play the kind of role that we think it plays, according to a lot of research that's come out in the last decade. One interesting study was from Wilhelm Hoffman and his colleagues, who basically gave people a device to carry around. They did this about 10 years ago. I think they were actually carrying BlackBerry devices. And basically, several times throughout the day, the device would buzz at them and say, basically, have you experienced any desires recently? And if they say yes, it would ask them further, did you try to resist the desire? And if they say yes, were you successful in resisting the desire? So you would think that if willpower is really important for sort of behaving in the world according to our goals, then people who claim to have better self-control, so we can ask people questions like, do you make impulse buys at the grocery store? Do you say things without thinking? things like that, and use that to basically rank people on, how good is their self-control out in the world, you'd think that those people with good self-control would be like, more often kind of, tamping down their desires, right? And doing things that they want to do rather than doing the things that they think they need to do rather than they're, sort of indulging their immediate desires. But what Hoffman and his colleagues found was actually sort of the opposite, that people who claim to have, high degrees of willpower or self-control actually seem to have fewer desires in general. And they don't seem to differ in the degree to which they are actually successful in overcoming their desires. They're just as successful as the people who claim to have poor self-control. So there's this kind of growing idea that, willpower is really more about putting oneself into situations where you're kind of avoiding your desires in the first place, rather than necessarily being about the ability to kind of squash our desires as they happen. So this kind of misconception about willpower highlights a more general problem, which is that we really need a new science of behavior change. And one of the arguments I make in the book is sort of in service of why it is that we need this. And part of it has to do with the failure of our ability to help people change their behavior. This is we call these abstinence plots, where basically it shows the percentage of people starting from the day that they go abstinent, showing how many people remain abstinent over the course of a year. And so what this shows is that, in 1971, if you look at alcohol, heroin, and smoking, about 20 to 30% of people were able to stay abstinent for a year. Fast forward to 2011, and you see basically those numbers have hardly budged. I don't have smoking numbers on here, but they aren't really any better now either. So, the people who have been doing, there's a ton of science has been done in the last 40, 50 years on these types of questions, but they haven't really gotten us anywhere. And we think that in part that's because it's been the wrong kind of research. So if you look at the theories and models of behavior change, they look kind of like this. This is probably the best known model. It's called the trans-theoretical model, or the stages of change model. And basically what it describes is like the process that a person goes through in trying to change their behavior, starting to kind of pre-contemplation, and then thinking about it, and then trying to do it, and then ultimately trying to maintain it and hoping they don't relapse. The problem is that this doesn't really tell us anything about kind of why things are happening, about what the mechanisms are in the brain or in the world that are related to basically why somebody might be successful or not. So you can think about this as sort of like a theory in medicine that, like, let's say we had a theory of, heart attacks that was all about the progression of symptoms of heart attacks. It's not telling us anything about where it actually comes from. So many people have been arguing that we need to take basically take inspiration from other areas of medicine where understanding mechanisms has been really key to improving health. And this is probably like the flagship example of that. There's a disease called CML. It's a type of leukemia that before the development of this particular drug, Gleevec, if you were diagnosed with that disease, you had about a 30% chance of surviving for 5 years. Now, it was discovered, quite a while ago that almost everybody with this particular type of leukemia has a very specific genetic mutation. It's basically, these bits of these two chromosomes get kind of mixed around, and it causes a problem with these couple of particular enzymes. So somebody came up with a way to basically develop a drug that can block that enzyme problem. And basically this drug on its own took survival from that disease from 30% up to almost 90%. And so this highlights the fact that when we understand these underlying biological mechanisms, it provides us the really powerful lever into figuring out how to fix things. So in the book, I talk about 3 different types of potential mechanisms of behavior change. One is thinking about the environment and changing the environment. for example, there's some really interesting research showing that if you really want to change your habits, one thing you can do is move. That when people move, they're much more likely to sort of be able to change habits that they want to change. Another thing we can think about doing is weakening habits, and I'll focus on that in a moment. And finally, we can think about sort of strengthening self-control. There's a lot of ideas around, say, meditation and other types of training. As I talk about in the book, the evidence for all of those is still, pretty minimal. So we don't really have good ideas about how to strengthen executive control particularly well just yet. So I want to focus on the question of whether we can hack our habit system. Can we go in and do something to actually change habits? And one clue comes from this phenomena that was mentioned in the trivia, where a small number of people who've been followed by Antoine Bashara and his colleagues, after a stroke, will basically stop smoking. These are people who might have been, hardcore smokers for years. And this is a quote from one of their patients. He said, I just woke up and I forgot that I was a smoker. No longer had the urge to smoke. And so this provides kind of a proof of concept that, it is the case that a change in the brain can get rid of a habit. Now, obviously, a stroke is a very dangerous thing. You don't want to go cause strokes just to help people stop smoking. This happens in this area called the insula, as we saw in the trivia. And the insula does lots of things. It helps, regulate lots of different physiological functions, for example. So you wouldn't want to just go and damage it. Nonetheless, it suggests that habits do live in particular parts of the brain and they can be disrupted. So this raises a question of, whether specific memories could be erased. Obviously we can erase memories on computers. An analogy that I talk about in the book is something that happened to me a few years ago where I had a bump on my forehead, kind of like a little pimple, but it never went away, kept getting bigger. And so I finally went to the doctor and they said, oh, you have skin cancer. Not a particularly bad type, but you have to get it removed. So I go to the surgeon and she cuts a little hole in my forehead and takes it out, sews me up. A couple months later, you can't even see any evidence that I had any surgery. So, what if we could surgically remove memories in the same way that we can surgically remove skin cancers? Well, it turns out that there's at least a couple of ways that neuroscientists have come up with to try to remove memories. And this is still, early days, but there's increasing evidence that it is possible to actually, kind of almost surgically remove a memory. So I'm going to tell you first about a way that it can be done with a particular drug. And the way that this was demonstrated was sort of interesting. So many of you have probably had this experience where you eat something and then you get sick. And then, for the rest of your life, you just can't stand the smell or taste of that particular food. This is what we call a conditioned taste aversion. And it turns out that you can generate these in a rat by basically just exposing it to a new flavor. These occur much more likely if it's kind of a novel flavor. And then injecting the rat with this stuff called lithium chloride that basically just makes it nauseous. And so once you do that, the rat doesn't want anything to do with that particular flavor anymore. So let's say you do that and you cause the rat to have that aversion. And then you inject this drug called ZIP into its insula. The insula turns out to also be important for taste and for taste memories. And so if you inject ZIP into the insula, what ZIP does is it affects a particular thing in the brain, a particular molecule called a protein kinase, which turned out to be really important for forming new memories and maintaining memories. This affects a particular type of kinase called PKM zeta. This thing is really important for kind of sustaining memories. If you do that, you basically get rid of the taste aversion. The animal will now go back and eat the thing that it previously, before the injection, wouldn't eat because it had the taste aversion. Now, ZIP is not exactly something you want to just go injecting in your brain. It actually, can damage neurons, and obviously in general, sticking a needle in your brain is not something that any of us wants to do lightly. But nonetheless, it shows that it is possible, by chemically modulating the brain to actually remove specific memories. It doesn't affect their the other aspects of their behavior. It seems to be fairly specific to that particular memory. Another potentially more useful way of trying to manipulate memories comes from something that's been learned over the last few decades that's really interesting about memory. We often think that once we kind of, create a memory, then it's there and we might forget it, but, it's kind of stable once we create it, which turns out to not exactly be the case. So let's say that you create a new memory. Let's say that you came up to that fork in the road and you went left and you got the rainbow at the end of the road. In order for that memory to become stable in your brain, it has to be what we call consolidated. Things have to change over time. In particular, the whole machinery of creating new proteins has to turn on so that can basically be stabilized. And once that happens, then the memory is fine. It doesn't need any more kind of stabilization unless you get reminded of it. And it turns out that whenever you get reminded of something, it causes that memory to become unstable again. And it has to be what we call reconsolidated. The idea is like it becomes unstable again. And then that same process of consolidation has to happen again for it to be both, updated, but also to become stable again. And if certain things happen right after it becomes unstable, right after you're reminded of it, for example, if you're a rat and you get injected with drugs that turn off your brain's ability to make new proteins, or if you suddenly learn that, hey, you don't get the reward you used to get, that can actually cause that memory to get lost. And so people have become increasingly interested in whether we can use this to actually kind of get rid of memories. There's an interesting, sort of anecdote that I talk about in the book of a physician back in, I think, the 1960s, who was doing electrical, basically, shock treatment, electroconvulsive therapy on his patients, and would have them actually stay awake for the shock therapy. And when he did that, if he had them talk about their sort of, memories of abuse or whatever it is that was sort of haunting them, they would actually lose those memories when undergoing that shock treatment. And that's one of the types of things that might be able to actually, help get rid of memories. This is, again, science fiction right now. There's no way that this is going to be done. That hasn't been demonstrated in humans. Or sorry, reconsolidation has been demonstrated in humans, but it hasn't been demonstrated that we can actually use it to sort of change habits. But nonetheless, there's a lot of interest in whether this is going to be something that can be used in the future. Finally, I want to mention optogenetics, which we talked about in the trivia. This is, by far the biggest development in neuroscience in the last 2 decades. And it's basically a technology that allows neuroscientists to control brain cells very precisely using light. So what they do using various forms of genetic engineering is basically take these particular ion channels taken, for example, from green algae, that basically open up when they are shown light, or they close up when they open up to different types of ions when they're shown different types of light. And so using those, putting them into very specific cells, you can either turn the cells on using light or shut them down using light. This allows this incredibly precise control over the activity of brains largely right now in mice and rats and other, fish and other animals like that. Some really interesting research by Anne Graybiel and her colleagues at MIT has basically shown that by stimulating very specific connections between parts of the prefrontal cortex and parts of the basal ganglia in mice, they can pretty precisely turn on and off particular habits. These are habits around, like, running in particular places to get food. But it really provides a proof of concept that one day, it might be possible to actually turn habits on and off. Now, getting this into humans obviously would be very challenging, in part because it would require something akin to gene therapy, using viruses to place these ion channels into neurons. And there, obviously there's major risks in that sort of thing. So one would have to be struggling with a really bad, challenging habit in order to undergo brain surgery for this. In addition, there's a lot of work in mice that's already shown us exactly where in the brain these types of habits develop. We don't really know that in humans because we can't do that kind of research at the level of neurons to try to ask which specific neurons seem to be involved in these sorts of habits. But nonetheless, it's a really interesting, sort of future direction for the potential control of habits down the road. I want to end by, kind of musing about what the COVID pandemic has taught us about habits and behavior change. And I think one of the things that it's taught us is that, when the context changes, we can change our behavior pretty quickly, right? That at least in San Francisco, we went from nobody wearing a mask on the street within a couple of weeks to pretty much everybody on the street wearing a mask. And that speaks to the degree that people can change their behavior very quickly. On the other hand, now that, at least in California, and I think everywhere, we've been told that you don't have to wear a mask outside anymore. I certainly, did not have any problem discontinuing that behavior. It certainly had not become a habit, even having done it over the last year. And I'm guessing that, many people had that same experience. Simply doing the thing over and over was not enough to kind of generate a really, the kind of habit that would take work to overcome. I think, maybe more importantly, another thing that we've learned is that, the kinds of ideas that I've talked about the brain systems involved in habits and the stickiness of habits are just one part of the wider problem of behavior change, of getting people to change their behavior. And we've seen that in COVID, in the fact that there's these sort of beliefs and ideologies and mindsets that have driven behavior much more strongly, I think, than sort of, the kind of habits that I've been talking about. And I think that these types of sort of higher level beliefs and mindsets become increasingly important when we think about, how do we address the climate crisis, right? Clearly, the climate crisis is I think most people would agree, is like the biggest existential crisis to the species to date. And, and solving it ultimately comes down to behavior change, both individual behavior change and societal behavior change. And part of that clearly is us, overcoming our habits about wanting to be able to take a warm shower and, needing to eat the things that feeling the need to eat the things that we eat and so on. But another big part of it has to do with the mindset about, whether people actually feel the need to change, right? If you don't feel the need to change, then there's no reason to change your behavior. And so I think that ultimately our science of behavior change needs to incorporate both thinking about those very, high level things about mindsets and beliefs and ideologies and how they relate to behavior change, along with, how we ultimately make changes from the bottom up. So I want to thank you very much and happy to go to questions now.
Awesome. That was so interesting. Thank you so much, Russ, for that great lecture. So we're going to go to some questions now. So feel free, if you haven't already, to put some questions in on YouTube or on Facebook. We had a question about, you mentioned it earlier in the trivia, but can you talk about the 21-day to change a habit? Is there any validity in that? And where did he come up with that?
I forget the story of exactly where he came up with it, but basically he kind of made it up. There was no evidence behind that claim. There was certainly no research behind it. And the short answer is that there's no one number. There's some work that was done a few years ago that looked at people, followed people as they generated habits, and trying to generate new good habits. And they found that people varied from, and they looked at when did it sort of become habitual in the sense that they no longer needed to think about doing it. And they found across different habits it varied from 2 weeks to more than 2 months. And so it, the numbers are, sorry, more than 4 months. And so that, the numbers really vary depending. There wasn't enough data there to like systematically say, well, certain types of habits are easier to do faster than others. And it's probably also the case that, the context matters. Certain people are probably going to be able to, do them more than others. I mean, the challenging thing about good habits is, I talked about how bad habits, are generated so easily because of, the role of dopamine in kind of cementing habits. When I let's see, what's a habit that last year I started wearing a night guard because I was like clenching my teeth, right? So you have to remember to put it in every night, right? And it's not particularly unpleasant, but it's also not particularly pleasant. It's not like I get a reward every time I put it in. And it definitely took me several months to be able to do that. And in part, that's because I had to think every night about it, right? I had to remember consciously, oh, I need to put in the night guard, or have it in a place where I would obviously see it and be reminded of it, because I didn't have that kind of dopamine, the rewarding outcome from doing it that usually would generate a habit.
Mm-hmm. That makes sense. Excuse me. All right. So we had another question about how first behaviors are wired as the default. Does that imply that behavioral habits learned in childhood are generally harder to unlearn or to change?
Yeah, that's an interesting question. I mean, most of this work is not looked at. In fact, I don't know of any work that's really looked at this longitudinally, across development. And so the short answer is I'm not really sure, but there are good examples of, habits developed in childhood that do go away over time, right? I think, fingernail biting is a good one. Thumb sucking is a good one. And even like, a really common problem in children is Tourette syndrome, right? Where they have tics, which are, we actually think that tics are really kind of like the habit system kind of gone mad, right? The habit system overreacting and basically like taking something like, twitching your eye or whatever that should just be something you do once, but it becomes a habit that is sort of like that the child is like unable to stop, right? Mm-hmm. And most kids grow out of those tics. In some people, they persist into adulthood, but most kids actually grow out of them. And so that's an example of how some types of habits we grow out of. I don't know of any work that's actually, kind of gone into why it actually is that some go away and some don't.
Okay. Could that also be related to why you are sometimes find yourself acting like your parents or doing things like your parents do?
There's probably a whole other set of things like that. That's certainly part of it, because obviously, you learn routines from your parents, right? Mm-hmm. And both by observation and by kind of like, having them show you how to do things. But there's also genetic factors, you share genes and environments with your parents, and there's lots of things that can drive you to be similar to them. So.
Cool. Thanks. You talked a little bit like kind of cross about this, but how do you differentiate a habit from an addiction?
Yeah.
Is that like a technical thing, really?
Well, I mean, addiction, I talk in the book about how there's actually a lot of questions about whether addiction is really about habits at all, right? It's almost certainly not about like behavioral habits, right? So if you like so you look at like what a person goes through who's like, trying to obtain drugs, and you see some of the stories about the really clever things people will do to try to manipulate others to give them drugs. Clearly those aren't habits, right? What's the habit is really the desire for the drug. So it's almost like, addictions are kind of like, sort of like emotional habits where the habit is really the desire or the craving for the drug. So there's ideas, and clearly dopamine is critical for that. So the idea is that, one thing that happens in addiction is that, that an individual becomes sort of sensitized to the it's called incentive sensitization, basically sensitized to the value of that particular substance. Now, what's interesting, kind of going back to the point I made about kind of liking versus wanting, you know, when somebody's deep in addiction, they often don't actually like the experience of the drug, except to the degree that it like brings them back from feeling really bad, right? And there's a whole, you know, there's all these really interesting relationships with stress and addiction as well. But I think that, you know, there's been increasingly some argument. I think most people still think that habit plays an important role in addiction. But there's an increasing argument that we need to think about addiction more as almost like overactive, goal-directed behavior, where the goal is the drug. And there's still a lot of controversy about that set of ideas.
Cool, thank you. We had a question about, does sleep contribute to habit building and consolidating?
Yeah, there's certainly the best work on this, I think, has been looking at like motor habits, like almost like motor skills, like teaching people to like, you know, press a bunch of buttons in a certain order really quickly and things like that. And it's certainly been seen there that sleep is required for the optimal, you know, basically development of those habits. And I don't remember, you know, different types of learning and memory require different types of sleep. And I don't remember if it was like, you know, REM or non-REM sleep that's important for this particular type of motor habit stuff. But sleep is definitely, there are definitely things that go on that I think relate back to those, you know, when I talked about consolidation, the idea like, you know, memories take some time to kind of get sort of stabilized. And we think that a lot of what's happening during sleep is that sort of stabilization is going on. There are biological things that happen in the brain during sleep that kind of drive that.
Cool, great, thanks. We have another question about how does ADHD affect habits or the decision and habit system?
That's a really interesting question. I don't actually know of work that is kind of, I mean, I think that the jury is out as far as I know. I mean, there's been some work that's like compared like people diagnosed with ADHD and people, you know, and people who aren't diagnosed, showing some differences in their ability to learn skills, to like learn motor skills in particular. I don't know how well that's held up. And so I wouldn't want to make a lot out of it. I mean, I think, you know, one challenge with ADHD is that ADHD is a really complicated phenomenon. And we don't think that like most psychiatric diagnoses, you know, most of them are not biological things. They're not like one biological thing. Like clearly ADHD is a biological thing in the sense that something is going on in the brain, but it's not the same thing across everybody with ADHD.
Right. Yeah, it shows up in different ways.
And so it's you always have to, when you see research talking about people with ADHD, you have to keep in mind that it's really mixing together probably a bunch of different types of people who, I mean, you can be diagnosed with ADHD in many different ways, right? So.
Yeah. All right. How can thinking patterns be habits?
Yeah, definitely. And I think this is, you know, at the extreme, something like obsessive compulsive disorder is basically like, you know, habitual thoughts, right? Thoughts that are evoked by particular stimuli out in the world that are uncontrollable. Yeah. So just like emotions can be, I think you think of like, you know, phobia or things like that as kind of like emotional habits, or even obviously, you know, even in, you know, I think, and this is kind of veering pretty far away from the science, but if you think about people in relationships, right, that people over time develop emotional habits where like, you know, their partner will do some particular thing that causes them to have a particular reaction that they may have trouble controlling. And that might be an emotional reaction, but it might also be a thought. Yeah. So I think that in some sense, you know, anything that our brain can do can become habitual. And it probably works, you know, for example, I think, you know, like fear works in a different way than sort of like motor and cognitive habits, because there are specialized systems in the brain for fear. But I think, you know, I don't think we know about these others, whether they exactly work in the same way, but they can definitely become habits. Yeah.
Great, thanks. We had a question, how do we change emotional habits, such as the ones you referred to for addiction or ones that might be leading to depression?
Yeah, interesting question. I mean, I think that ultimately it comes down to, figuring out how to like because habits, because our executive control and our willpower is generally so weak in comparison to the habit that like once the habit gets turned on, it's really hard to turn it off, right? So part of it is about either just like figuring out how to avoid the triggers for the habit. This goes back to that idea of like, changing the environment. Like, can you avoid the triggers, or can you somehow sort of reconceptualize the triggers so that they don't kind of trigger it in the same way? And that's like, for example, in cognitive behavioral therapy, what one does is kind of like rethinks your reactions to things, right? So the idea is like, can you short circuit the habit before it ever gets turned on?
Kind of interrupting it, taking a turn before you come to that first why.
Exactly. Yeah.
Yeah. Is dopamine fasting sensible biologically, or what's going on there? I was not aware of this trend, so I don't know about it too much personally.
Yeah. So the idea is like, it's one of these like classical Silicon Valley, kind of pseudoscience-y, snake oily type things that, that come out occasionally. So the idea was basically like, it basically is, it's like a modern day version of a meditation retreat. The idea is like, get rid of all stimulation for a week, and like, let yourself like, let your dopamine system calm down, right? That's the idea behind the dopamine fast. There's no science behind it at all, right? In part because your dopamine system is always going to be doing its thing. And if it weren't, like I said, you'd be catatonic. You need the right level of dopamine in order to be kind of, on. And like too little, you'll be, really dull or ultimately like passed out. Too much, and you basically become frazzled and, anxious and freaked out. So there's this kind of like Goldilocks zone in the middle of like, you want the right amount of dopamine. And your brain is generally, pretty good at kind of figuring out where that is, unless you either do things like throw cocaine at it, or you get really stressed out, in which case, like, a lot of dopamine gets thrown out. So the whole idea that like, putting down your phone for a week and, it may well be a good thing to do. I'm not saying that it's not, a lot of people have good experiences of meditation retreats. Some people don't, right? Because not talking for a week turns out to be a really hard thing for some people. I couldn't do it. I've never tried it, but I can't even imagine trying it. But it has nothing to do with like turning down your dopamine system.
Okay. I assume they also don't serve bananas at these places.
That's an interesting question. I don't know if the dopamine in the banana actually like survives digestion or not. Okay. I'm not really sure. Yeah.
Yeah. I wonder do you know if there's any purpose? I mean, why would a banana need dopamine? That's a whole different question.
Yeah. I mean, apparently, dopamine is a really fundamental chemical for lots of different things. Like, dopamine they use in emergency rooms, right, to like stabilize people's blood pressure and stuff. It's, not because it's affecting their brain, because it's affecting like the dopamine receptors in their blood vessels. Yeah. So it's something that like, I don't actually know like what it's doing in vegetables, but it's there in vegetables.
All right. Okay. We had a question that was, is compulsive behavior an overdrive of some part of the dopamine system? You kind of talked a little bit about this. But
I don't know of great evidence that shows, sorry, I sound cagey. I don't know of great evidence that shows that to be the case. Some people have reported that, but I think it's one of the problems with dopamine is that it's really hard to measure in humans. The only way you can directly measure dopamine is by injecting people with a radioactive substance that binds to the dopamine receptors. And then you can do that to measure it. But, you can't do it, like a person can basically only do that like once a year, and they've had their maximum, radioactive exposure for research. And it's also really expensive. It costs like thousands of dollars per person because you have to have a chemist making up these chemicals for each time. And obviously it has to be very safe and regulated. So I think the short story is, I would guess that dopamine almost certainly has a role, but I don't think we know what it is. Yeah.
Yeah. I understand your caginess. Sometimes we when you don't have the answer, you can't give the answer.
Exactly.
Yeah. Another question is, are the mechanisms that enable someone to learn physical skills, such as playing an instrument, similar to the mechanisms you've talked about today?
Yes, definitely. In fact, I think a lot of what we've learned about the quote unquote habit system in the brain actually comes from studying people learning skills, be they, motor skills, like I mentioned, like learning, to press particular kind of like a oversimplified version of like learning clarinet or something, like pressing, certain buttons, or like stuff that I've studied a long time ago is like training people to like, do new perceptual skills, like training them to read words that are shown through a mirror, right? Which it turns out that over time you can get better at doing. And, we can look at how those things work. And I talk about this in the book, how the basal ganglia seem to be important for all of those types of skills.
Cool. One more question was, could you comment on the use of beta blockers for treating PTSD?
The short answer is it's too far out of my area. I don't know the research there. Yeah, so sorry.
Sure. That's okay. I was wondering a little bit about what your methodology is in your lab. Like, how do you go about doing your experiments? If you're talking about, you can't really measure dopamine very easily because that, yeah.
So we use MRI, magnetic resonance imaging. We put people inside an MRI scanner. They have, little goggles on so we can show them video. They have headphones, they have buttons so they can like respond to things. And we basically give them tasks to do while we're scanning their brain. And then what we do is, so let's say, for example, we give them a task where they have to make choices and occasionally they get like a monetary reward, right? And we want to know, how is that represented in their brain? What kind of changes does that cause? So then what we'll often do is basically come up with kind of like mathematical models that tell us like, what would we expect if a part of the brain, so for example, let's go back to this idea of reward prediction error, right? The idea that like, dopamine neurons respond to either to unexpected rewards or to unexpected stimuli that signal that a reward is coming at some point in the future, right? So we can come up with a computational model that tells us like, what should, at any point in time during our experiment, what should the level of reward prediction error be that the person is experiencing? And then we can ask, where in the brain does activity move along with that? So we can kind of like use these statistical models to ask, what parts of the brain seem to act like they're doing particular types of things.
Cool. So it's not necessarily like rat research for you.
No. So my lab doesn't do, I talk a lot about rat research because we've learned a lot from it. My lab only studies humans. Yeah. So we can't image dopamine directly, right? We're imaging, when we do what we call functional MRI, we're imaging basically blood flow in the brain, which changes in response to neurons firing. It also changes in response to dopamine. We can't really tell the difference. But we're certainly seeing the effects of dopamine, but we can't pull that apart from other types of neurons being active.
Okay. Another question is our general dopamine level genetic? Is there anything we can do to alter it, such as food or drink or diet?
There certainly are differences between people in, for example, there's a set of enzymes that basically kind of suck up dopamine in the brain. And like, basically the dopamine neurons spit it out, and you wouldn't want it to just sit around forever because it would sort of build up. And so you have these enzymes that basically take it and break it down back into its like original parts, and then it can get reused. And those, like, different people have different versions of the genes that create those enzymes, and some of them are kind of more powerful than others. It's not really clear that those differences actually make a big enough difference across people to matter. There's, I talk about this in the book, how there's, like 10, 20 years ago, there was a lot of research, saying, well, this particular gene, if you have this version, you're going to have like better executive function than this other version. None of that research is held up now that we have like these really big studies that look across the entire genome. We think that it was really kind of like research cherry picking, if you will. And so it's not clear if those do make a difference. They make a really small difference. Now, whether you can change your dopamine level, other than like doing cocaine or methamphetamine, which I would not recommend. I don't know of, like, there, it may well be possible to change it. And I don't know that you want to change it necessarily. I mean, I think that I guess the place where you would want to change it is like, if your general amount is like too low or too high, right? Then that would either make you like, too sedate or too freaked out. Now, in some sense, this is what like a lot of the drugs that are used to treat ADHD do, right? I mean, one of the most common drugs for ADHD is basically amphetamines, right? And what they're doing is increasing the amount both of dopamine and of noradrenaline, and a related neurotransmitter. And there's long been ideas that like, one of the things that happens in ADHD is that there's kind of a general decrease in the overall level of dopamine. I don't know that that's held up that well. But those drugs are really, all of these drugs that affect neurochemicals are really complex, in part because the neurochemicals are all in this really complex web. If you change one, it's going to cause like 5 others to change. So it's really hard to kind of pin down exactly what the effects are.
Okay. Just a quick question. Does cocaine or methamphetamines contain dopamine that your body processes, or is it just like release all of your dopamine really fast?
What they do, I'm trying to remember which one does which. Basically, what they do is primarily affect so there are these enzymes that are not enzymes, these little sort of molecules that basically like suck the dopamine back into the neuron. That's another way that dopamine's gotten rid of. And what they do is actually either stop those things or actually reverse them. So instead of like sucking in dopamine, they actually start spitting out more.
Oh, okay.
So they have sort of pretty specific effects on the dopamine neurons, but they're doing that by like changing the ability of the neurons to like, kind of regulate the amount that's sitting around.
Okay, cool. I wasn't sure, since you said the banana stuff probably gets digested. I wasn't sure.
Right.
Thanks. Another question from the audience: would we get desensitized to increasing dopamine background levels?
That's an interesting question. Probably, I mean, in the sense that the brain can get desensitized to almost anything, maybe. But the brain also has really powerful, kind of means of keeping itself within a good operating range, just like our body does. And so it will, in part because like, if things get too far out of whack, then, basically just the brain just goes haywire and you end up with like epilepsy or, something else like that. So everything has to be really tightly controlled for the brain to work at all. So I don't, so I think it's hard to say. Yeah.
Okay. So if anyone else has more questions, please put them in, because otherwise I might be asking our last question, which is a personal, selfish question. In some ways, I saw you had a cat. I also have a cat, and you were talking about prefrontal cortex being pretty small in cats. Is that maybe why they're so hard to train? Because it's hard to create new habits for them? Uh-oh. Looks like you froze, Russ. See if you can come back. Unfreeze. There you are.
Yeah, sorry, I missed
don't know if it's my internet or your internet.
I'm glad the internet flakiness waited until the very end.
Yes, yeah. All right, looks like you're there. I asked, is the lack of prefrontal cortex for cats possibly why they're so hard to train? So hard to like develop those new habits?
Almost certainly, yeah.
Yeah. Okay.
Yeah, they just, this is what I like about cats actually, but that they're very sullen and just kind of do their own thing, right? And I think obviously it's hard to know exactly why they're like that, but I think an important part of why it is that cats are impossible to train does have to do with that lack of prefrontal cortex.
All right. I will add that we did have a science pub with someone who does train cats, and they seemed to be successful, but it seemed like a lot more work than I was interested in doing with my cat. And I don't know if she would cooperate quite as well. Oh, so we've got a couple more questions that came in from the audience. One is it dopamine or serotonin or other chemicals that contribute to the feeling of happiness?
The short answer is I don't know. Well, let me say, it depends on what you mean by happiness. If you mean pleasure, then I think there's some cool work by this guy, Kent Berridge, and his colleagues from Michigan, where he's the person who came up with this whole idea of like a liking versus wanting. And people used to think that dopamine's all about pleasure and reward. And his research, along with others, has basically shown that story is not right. So what they do is they videotape rats like in their cage, like doing stuff, and then they go through and count how often they do. There's particular types of either what they call hedonic reactions, either positive reactions like licking their lips or licking their paws, things like that, or negative reactions like shaking their head. And these are reactions that are shared across all species, right? Human babies. He has some great videos like human babies, monkeys, rats. They all do these like things like flailing their arms when they're unhappy. And basically what he shows is that dopamine, if you like give the animal a drug that blocks dopamine, it doesn't actually change the degree to which they exhibit those kinds of like those positive or negative hedonic reactions. The neurochemical system that's involved in those kind of reactions is actually the opioid system, right? The target of heroin and morphine and so on. And his group has worked out what they call these hedonic hotspots that are in different parts of the kind of deep part of the brain that do seem to be involved in this kind of like pleasure response. Dopamine. So what happens if you block dopamine? Well, basically the animals still like stuff. They just won't work to do it. They just won't work to go get anything. They basically become like couch potatoes.
Okay. Interesting. Another question we got was, how do we change habits deeply entwined with our family or culture?
That's really hard, right? Because you need one of the important ways to change a habit is to change the context and change the environment, right? And if it's something that's like strongly supported by your environment, then that just makes it that much harder to change. So I think that, unfortunately, that if it's something that you really want to change, I think you have to like get away from that environment for a while.
Yeah, that's the tricky one.
Yeah.
A question going back to kind of related to my cat question, is the prefrontal cortex percentage of the brain similar in humans to other animals? Because the animals have a smaller area, but they also seem to have smaller brains.
Yes. Yeah. So it's certainly, humans have, apes have the highest percentage of all animals for sure. Yeah. And apes differ a lot in their size, right? There's tiny ones, there's big ones, but yeah, if you look at the percentage of, and that's a really good question. That's a good point. The percentage is the highest in apes. There's been debate about whether like humans have a much higher percentage than say chimpanzees or, baboons or other apes. It kind of seems, my reading is that actually, it's not clear that humans have that much more than, say, a chimpanzee, but it's organized differently. And the argument now is that, the differences between humans and chimpanzees, for example, probably have to do with very fine-grained differences in the prefrontal cortex.
All right. Well, I know you have a cat, but do you know anything about dogs? Like, do border collies and dogs that breeds that seem to be like really eager to learn have higher or different prefrontal cortex, or is that?
Sorry, I'm useless on that one.
Yeah, I can understand. Your research is not exactly animal focused, but well, it is because we're animals, but not on border collies. That's something that our audience will have to go and investigate themselves.
Exactly.
Well, I think that you answered all of my questions. I'm looking to see if I have any other questions that I had. And it seems like our audience has asked all the questions that they are going to ask. So I just want to thank you so much for coming and answering all of those questions for a really, really interesting lecture. Your book is out now and available for purchase. Hard to Break: Why Our Brains Make Habits Stick. And that is, I believe that we have a link and a discount available in our chat. So thank you so much again for coming. I really, really appreciate everything. It was really fun. Have a lovely night. All right. Okay, folks, we are out of time. I hope that you enjoyed tonight's event. If you would like to watch the video again or share it with your friends, you can check out the video section of OMSI's Facebook page or our YouTube channel. And don't forget to follow us on social media channels for updates on future events and inspiring content from OMSI. We're always putting out new stuff. I know I mentioned it at the beginning, but please consider supporting Science Pub and making a donation via the Facebook donate button, or you can visit omsi.edu/donate. Every bit helps. Join us for our next virtual Science Pub on May 18th. I believe that's next Tuesday. We're going to have a lecture by Paul North, who is the founder of Meet the Ocean. And he will take us on an exploration of the function and fashion of the great Alaskan marine invertebrates. So I'm assuming he'll be talking about those giant crab arms. Once again, thank you to our partner, Celestream, for helping make tonight's event possible. We couldn't do it without you. And as always, you can get more information on our website at omsi.edu.